Hello everyone. Today we are going to talk about hormones that regulate our blood cell production, and we are going to talk about the coagulation process. Thrombopoietin hormone is produced in our liver and it regulates our platelet production. Our platelets are pretty important in clotting our blood when we are injured. There's another hormone known as erythropoietin, and it's mainly made in our kidneys. This hormone controls our red blood cell production. We are going to talk about what stimulates the production of erythropoietin in a minute, but the process of producing those red blood cells by the hormone erythropoietin is known as erythropoiesis. Poiesis means to make. Our erythropoietin hormone produced by our kidney stimulates the production of our red blood cells. Our red blood cells are known to carry a protein known as hemoglobin, and that's the protein that's going to help your red blood cells transport oxygen. And so several things can trigger the release of erythropoietin. One is if you are low in oxygen. Another is if your numbers of your red blood cells are pretty low, or if your hemoglobin levels are low. All of these are going to trigger the production of erythropoietin by your kidneys and your liver. Once erythropoietin is released, it's going to act on your red bone marrow in your bones, because this is where we have the stem cells that are going to produce the red blood cells for us. And so this is one way how we can react to an imbalance that we face so that we can go back to that homeostasis level. This is how one hemoglobin looks like. This hemoglobin protein is the protein found in our red blood cells, and this is the protein that's going to help us transport oxygen. This hemoglobin protein is made out of 4 protein chains. We've got 2 globin chains and 2 alpha chains. In the middle of each chain there is a heme disc, and this heme disc contains the heme pigment which contributes to the red color of our blood. In the middle of each heme disc, or you can refer to it as a heme group, there is an iron atom in the middle, and this is where oxygen is going to bind. Iron is also needed to produce hemoglobin for us, and to make those heme groups we actually need iron. As I said, iron is needed for hemoglobin synthesis, and we get our iron from foods such as beans and spinach. Spinach is a rich source of iron. Once we ingest the iron, we are going to absorb it by our small intestine, and then it gets transported in your blood by a protein known as transferrin. It gets sent to the bone marrow, and this is where it's going to contribute to the production of our hemoglobin. Now, if we ingest too much iron, it gets stored in our liver, but we actually have to be aware of how much iron we are consuming, because we can get iron poisoning. And this iron toxicity can actually lead to the corrosion of your digestive epithelium, which is going to lead to internal bleeding and liver failure. Excess iron can get stored in our liver, and it gets stored inside of a protein known as ferritin. But as I said, too much iron and it's going to lead to problems. Now, our spleen is known to destroy our old red blood cells. Our old red blood cells only live for about 120 days. Then those red blood cells are going to be destroyed by our spleen. And when we break down our red blood cells, we are basically going to break down our hemoglobin. So we break down the globin chains, we break down the heme discs, we get iron. So those globin chains, when they are broken down, since they're made out of proteins, we're going to get amino acids. Now, those amino acids, we can actually recycle them. So we can actually take those amino acids and incorporate them in making new proteins. The iron is going to also be reused to try to make new heme groups. Our spleen and our liver as well can convert some of those heme discs into a pigment known as bilirubin. Bilirubin is usually carried by your plasma protein known as albumin, and it gets sent off to your liver. Your liver is going to metabolize bilirubin, and it's going to secrete those metabolites with the bile, which is released out of your liver. And also, bilirubin is going to, the metabolites are going to leave your system in feces, along with your feces coming out of your intestine. Bilirubin metabolites are a small amount of them are actually going to also be released out in your urine, and it contributes to the yellow color of your urine. And also, the bilirubin contributes to the brown color of your feces. Liver disease, if someone has a liver disease, um, that's going to make it hard for the liver to actually process and excrete the bilirubin, which can lead to jaundice, which is the yellowing of your skin and your eyes. When we are injured, to heal our wound, we usually go through three steps: vasoconstriction, and then the second step is a platelet plug formation, and then the third step is coagulation, and that's the step that we are going to talk about in detail. The first step, though, vasoconstriction, when we injure ourselves, we actually um, decrease the blood flow within the injured vessel by constricting it temporarily so that we can allow time for the repair process to proceed. The second step is a temporary blockage of the break by a platelet plug. So a bunch of platelets are going to rush to the injured scene, and they're going to adhere to the damaged area. And it's going to create a platelet plug. The third step is coagulation, where we are going to form a clot one more time to seal the break until your tissues are ready to repair themselves. So the clot is just a temporary fix until we start healing. The protein fibrin is going to as you can see right here, kind of create that mesh look right there, and that's going to help stabilize the platelet plug that was previously formed, and that's going to help form a clot. So that again, we can stop our bleeding and we can start the healing process. Now, how do we get the fibrin protein? There is an enzyme known as thrombin. This enzyme helps convert an inactive form of a protein that we have known as fibrinogen. It helps convert fibrinogen into its active form known as fibrin. Now, whenever you see usually the suffix gen, that would tell you that this protein is inactive. And for this protein to go ahead and get to work, it needs to be activated. And so the enzyme that will activate fibrinogen for us is the thrombin enzyme. Now the fibrin fibers, as you can see right here, they're going to weave themselves through the platelet plug and they're going to trap the red blood cells within the mesh. As I said, the clot that we're making right here is just a temporary fix. Once your tissues start to heal, we would want to break down and disintegrate that clot. And so to do that, we need to go ahead and start breaking down the fibrin. Now, to break down the fibrin, we need the help of an enzyme known as plasmin. Plasmin is going to break down the fibrin and therefore break down the clot. Now, to get the plasmin enzyme, thrombin and a tissue plasminogen activator, TPA, they go ahead and they convert an inactive protein known as plasminogen into its active form known as plasmin. Once we get plasmin goes ahead and breaks down fibrin, and that process of breaking down fibrin is known as fibrinolysis. Again, lysis means to break down, and so we are breaking down fibrin. And this is how we break down our clots. Now, we have to make sure or we have to be kind of like aware of how much of the thrombin enzyme we are making. If there is a disorder and someone is making too much thrombin, that would be a problem because production of too much thrombin is going to increase the production of fibrin, and that's going to increase the clots in our blood, and too much blood clots, and that can block our blood vessels and lead to so many problems. Another disorder that we also need to be aware of is that some individuals produce very little of these enzymes, thrombin and other, and those like right here, TPA. And so, therefore, the problem could be is that they are not going to be able to make enough of those clots, and so therefore, when they are injured, their blood is going to be it's going to bleed profusely because they are making very little amounts of those clotting factors.